Hello students, welcome to the discussion of theory paper for 2025-26 according to the PU board, and this is about the PU board theory paper which was given this year, that is model paper 2. So let us start with first MCQ question. Statement 1. Apomixis is the production of seeds from unfertilized ovules. So now here you have to choose which one is the correct statement among this. So apomixis is the production of seeds from unfertilized ovules. We know the definition that apomixis is production of seeds without fertilization. So the word unfertilized refers for without fertilization. So the statement 1 is correct. And the statement 2. Embryos produced from apomictic seeds are not generally identical to the parent plant. See here, apomixis, usually there is no fertilization. When there is no fertilization, automatically the parental character will exactly appear in the offsprings. So the embryos will always be generally identical to the parent plant. They have given not identical. So this is wrong statement. So according to this, statement 1 is correct, statement 2 is wrong. So the option is first one. So let's go to the second question. The cells that secrete testicular hormones, androgens are. So now we know seminiferous tubules are lined by Sertoli cells and male germinal cells. So in between these Sertoli cells and male germinal cells, you can find Leydig cells or interstitial cells which are meant for secreting testicular hormones which are commonly called as androgen. So the correct option is Leydig cells. Which of the following is not a copper releasing IUD? So we know there are different types of IUDs. So intrauterine devices. So we have first one as copper releasing IUD, hormone releasing IUD, non-medicated IUDs. Among these, they have asked not a copper releasing IUD. So we have copper T, copper 7, multiload 375 as the example for copper releasing IUD. But LNG20 is the only one which is not the copper releasing IUD, but it is the hormone releasing IUD. So the correct option will be LNG20. What is the possible blood group of an offspring when both parents have AB blood group? So we have to find out what offsprings have been obtained from the AB blood group. So according to this blood group, we have a parents now AB with AB. So what will be the offsprings? So the possibilities, one is A blood group, the other one is AB blood group, and you can go for other combination with the B blood group. So we have totally A blood group, B blood group, AB blood group in the offsprings. So the correct option will be the B. Next question. In mRNA, which runs from 5' to 3' direction, in translation, how many types of amino acids are present? So if you want to find out the amino acids, we know that genetic code property, always genetic code is triplet. So we have first one, it is read in the form of 3 nucleotides. So the first one which is coding for methionine amino acid, and the next 3 you can take like this, they are all same. So these will code for phenylalanine. So we have last one code here which is stop codon. So we have UAA or UAG or UGA. They do not code for any amino acid. So now they are asking how many types of amino acids are present. So we have one type and this is another type. So totally the answer will be 2 types. Arrange the following animal groups in the correct sequence according to zoological time scale. So according to the zoological time scale, we know that fishes are the first, then they gave rise to amphibians during the course of evolution, then reptiles, then comes mammals. So the correct answer will be 2, 1, 3, 4. Antivenom given against snake poison contains. So this comes under immunity. So in that, the subtopic is immunization. So we know that whenever snake bite is there, so we give ready-made antibodies. So this will be the correct answer. So first one is antigen, it is opposite to antibody. So antigen antibody complexes are not given, enzymes are not given. The preformed antibodies are given during snake poison. The substance produced by a cell in the viral infection that can protect other cells from further infection is, see already a cell is there which is infected by a virus. So if a cell is there, if it is infected by virus, there will be other cells which are present beside. So these cells will secrete chemicals which are called as interferons, which come under cytokine barriers, and they will give alarm to the other cells that further there should be no viral infection to that cell. So thus it gives protection from the infection from bacteria. Because of the presence of the following vitamin, curd is considered nutritionally richer than milk. So this is vitamin B12. So it enhances the nutrition whenever the milk is converted into curd. Mycorrhiza is involved in absorption of, this is symbiotic association. So the roots of gymnosperms are having association with the fungi, where this fungi will absorb phosphorus from the soil and passes it to the plant. In turn, they give the plant will supply the fungi nutrition to the fungi. So phosphorus is supplied by the mycorrhiza. So there is PBR 322 vector diagram given. Identify the site A, B, C, D. A site, B, C and D. So according to this, so A site is the ori site which is required for replication of this vector, and B is ampicillin resistant, and C is tetracycline resistant, and D is HIND3. So according to the diagram of PBR 322, you can have these labelings. So the correct option is B. The plants developed by the fusion of protoplast of two different plants are called. The tissue culture, we learnt about this. The two cells, so the cell wall is digested. And later on this protoplasm is fused, and this is called as somatic hybrids. Example, we have tomato, the protoplast of the tomato fused with the protoplast of the potato, which we get pomato. In a pond, if there are 20 lotus plants last year, so through reproduction we have seen the 16 are added. Calculate the birth rate. So they have asked birth rate. So what you have to do? Divide it by 20. You get 0.8. So 0.8 offsprings per lotus per year is the correct answer. The correct sequence of steps in the process of decomposition is, so we already know decomposition. First there will be fragmentation, then leaching, catabolism, humidification, and finally there will be mineralization. So the correct option is A. The last question. The following are the in situ conservation approaches except, they have asked except which one? All are in situ conservation. So we have national park, wildlife sanctuaries, biosphere reserves as in situ conservation, and this is ex situ conservation. So the correct answer is D. Now we come to fill in the blanks. So the first one, the glandular layer of the uterus that undergoes cyclical changes during menstrual cycle is endometrium, which undergoes cyclical changes during menstruation. The paleontological evidence for evolution is obtained from, of course it is from fossils only. Then dash is very effective sedative painkiller, morphine. The plasmid DNA act as vector to transfer the piece of DNA attached to it. The genetic diversity shown by Rauwolfia omichoria growing in Himalayan ranges produces an active chemical called reserpine. Now we come to the part B. So 2-mark question. The first one, write the significance of sporopollenin. So we know sporopollenin is present on the pollen grain. So when you see the structure of pollen grain, so outside there is sporopollenin, exine, and then there is intine. Then after that you can find the plasma membrane, then 2 cells, one is vegetative cell, the smaller one is generative cell. So this sporopollenin, what is the use? So pollen grains are well preserved as fossils because of this sporopollenin, because it is highly resistant to enzymes, alkali, acids. So this is preserved as fossils. The second advantage of this sporopollenin is it shows fascinating array of patterns which helps us to identify different plant species based on this one. And also this exine, which is made up of sporopollenin, protects the pollen grain from desiccation. So this can be written for 2 marks. So only 2 points are required. You can go for this. Mention the reasons for infertility in humans. So the infertility in the humans may be due to physical, congenital, or any diseases, due to drugs or immunological or even psychological problems. The person suffers from infertility. So give the differences between linkage and recombination. So whenever we speak about linkage, so on the chromosomes, if this is a chromosome, so where the genes are located, so we call it as linkage. We can say physical association of genes on a chromosome is called as linkage. Then recombination is when 2 chromosomes are there, parental chromosomes, there will be crossing over during meiosis, where there is exchange of genetic material, and this process is called recombination, the generation of non-parental gene combination of genes due to crossing over. So parental chromosomes, they get to overlapped on each other, and there is a point called chiasmata where crossing over takes place, and that is called as recombination. So there is a diagram based question. Draw a neat label diagram of nucleosome and label the parts. So as it is for 2 marks, 4 labeling is required, 1, 2 and 3, and this one can be 4. Comment on the similarity between the thorn of bougainvillea and tendril of cucurbita with reference to evolution. So there is thorn of bougainvillea, tendril of cucurbita. When you take these 2, thorn as well as tendril, so both are having axillary position, where you can find a leaf here. As these 2 are present in the same position, but they are having different function. One is having protection from the grazing of the animals, the other one helps for climbing. So they are basically, we can call them as they are having same origin but different function. What are flocks? Write the significance in sewage treatment. So during sewage treatment, there is a primary treatment, secondary treatment. Primary treatment includes physical removal as well as sedimentation. So secondary treatment, the masses of bacteria are allowed to grow in the aerobic tank, where we can call it as a large aeration tank, So when air is pumped into the sewage water, so the bacteria and fungi grows on that water body by using organic matter, and that structure is called as the organisms, which together we can call them as flocks. So it forms like a mesh, and they consume a major part of the organic matter from the effluent, and the significance of the BOD reduces during the growth of the flocks. So here they are asked what is flocks and significance, what is the use. Actually the flocks helps in reducing the BOD of the effluent. Mention the evil quartet of biodiversity loss in the given habitat. So biodiversity loss may be due to habitat loss, fragmentation, where the area is fragmented for the purpose of either cultivation or for development of city, over exploitation, alien species invasion, co-extinction. These all are very important, which will lead to the biodiversity loss, and they are the main cause. So you can write these 4, you will get 2 marks. And the diagram based question for 3 marks. Draw a neat label diagram of LS of embryo of grass. You have to draw this and you have to label at least 6 parts. 1, 2, 3, 4, 5, 6. Either 6. So scutellum, coleoptile, shoot apex, epiblast, radicle, root apex or root cap, coleorhiza. What is placenta? Let's its function. Placenta is said to be a temporary connection or association or bridge between the fetus as well as maternal tissue as well as blood. So it's a connection between the fetus and maternal tissue. It helps in transport. See, we have given many functions here. So for 3 marks, we need, this will be 1 mark, and at least you have to write 4 points. So it helps in transport of oxygen, water, electrolytes and nutrients from the maternal to the fetal, as well as it helps in removal of the waste, nitrogenous waste as excretory products, nutrients from the fetus to the mother blood, and it also acts as barrier for the entry of certain bacteria and harmful materials from the maternal blood. It allows the immunoglobulin antibodies to cross and provide the passive immunity to fetus. It also acts as endocrine tissue by producing some hormones like estrogen, progesterone, HCG as well as HPL. You can take the screenshot of this one and then use it for writing the notes. Draw a neat label diagram of Miller's experiment. So again you have to draw with 6 labelings. So Miller experiment with all these labelings, almost you have to mention the gases, boiling water, liquid water, and electrodes, vacuum pump, water droplets, condenser, etc. Define venereal diseases or STI. So the diseases or infections which are transmitted through sexual intercourse are collectively called as STD or STI. So mode of transmission, how STI is transferred. So it is due to unprotected sexual intercourse from the infected persons, as well as sharing of injection needles, surgical instruments, transmission of blood from an infected mother to the fetus. These are all the modes of transmission of STI. Mention the causative organism for the following disease: Ascariasis, Ascaris, typhoid, it is caused by Salmonella typhi. Filariasis, caused by Wuchereria bancrofti, Wuchereria malai. Name the microbes that help in the production of the following bioactive agents. We have bioactive agents like cyclosporin A, which is produced by Trichoderma polysporum, streptokinase, caused by bacteria called Streptococcus, statins, produced by a bacteria called Monascus purpureus. So represent pyramid of energy with appropriate units. It's a pyramid of energy and the unit should be mentioned. So sunlight, so how it passes from the 10% law, from the producers to the consumers, like that. So now we come to the 5 marks question. Explain the different outbreeding devices developed by flowering plants. We know that flowering plants avoid self-pollination and always promote cross-pollination. So here there are the steps where the flowers modify themselves to prevent self-pollination and promote cross-pollination. One of them is dichogamy, where there is bisexual flowers, male and female parts mature at a different time. There is no synchrony between the maturity of the male as well as female. Either the male will release its pollen grains before the stigma achieve receptivity, or the stigma achieve receptivity before the pollen grains are dehisced. The other one is declinia or unisexuality. Here the plants have only one type of flowers, unisexual flowers. Either they have male flowers only or female flowers only. So next is self-sterility or self-incompatibility. So here it is a type of genetic mechanism where the pollen grains of the same species or the same plant, if it falls on the same flower, it will not be allowed to germinate and produce pollen tube, and so it is called as self-sterility or self-incompatibility. So it's a diagram of mammary gland. Draw a neat label diagram of mammary gland sectional view and label the parts. So as it is for 5 marks, you need to draw it neatly and then label all the parts. Explain incomplete dominance schematically representation with inheritance of flower color, taking snapdragon as an example. So here you need to first write parental generation, which is capital R, red flowers, small r, white flowers. So capital R, we have gametes R, and white flowers produces small r. So when pollination is done, they produce capital R, small r, which leads to a type of flower called as pink flowers. So in the F1 you can find pink flowers are produced, and during F2 we do self pollination, wherein capital R, small r is crossed. During gamete formation, capital R, small r will be separated, and then we get the genotypic ratio and phenotypic ratio in the form of 1:2:1. So when you speak about phenotypic, there is 1 red, 2 pink and 1 white. Genotypic ratio, so the capital R is 1, capital R, small r 2, small r 1. So the phenotypic and genotypic ratio is similar here. So what is the definition of this incomplete dominance? Here the F1 does not resemble either of the two parents. Explain Griffith transforming principle experiment. So we are going to answer now what is transformation. So transformation is the ability of the organism to take a foreign DNA or genes from its surrounding through the cell envelope. So here Frederick Griffith conducted an experiment called transformation experiment. First he gave living S strain into the mice, and the mice, when S strain was injected, S strain has the capacity to develop disease, pneumonia in the mice. So the mice suffered from pneumonia and it died. When living R strain was injected into the mice was healthy because the R strain did not cause disease. When the S strain, which was causing disease, was heat killed and then injected into the mice was healthy, as this S strain lost the capacity of causing disease. When these both, living R strain and heat killed S strain, which did not cause disease, was combined and given to the mice, but the mice died. So the Griffith thought that something from the S strain which was heat killed transferred material to R strain living and transformed into the living S strain and caused the disease in the mice. So the live S strain bacteria were recovered from the blood. So in his experiment he thought that heat killed S strain transferred something that made R strain to become virulent S strain and caused the disease, and he thought that it was the genetic material which was transformed, and he called it as transforming principle. But he was not knowing which material has transferred from the heat killed S to R strain, which made the R strain to become S strain. What are the functions of I gene? We know that it codes for repressor protein, Lac-Operon concept. So I gene is responsible for coding repressor protein. LAC-Z gene is responsible for coding beta galactosidase enzyme. Y codes for permease. A codes for trans-acetylase, and they have asked one more is inducer. We know that during Lac-Operon the inducer is lactose. So this lactose binds to repressor protein and it forms a repressor inducer complex, wherein this will allow the genes to get activated. So the Lac-Operon gets switch on in the presence of inducer. Briefly explain the use of transgenic animals. So we have to explain how these transgenic animals are useful. So to study the normal physiology and development, these transgenic animals are very helpful as they are designed specifically for studying how the genes are regulated, how they affect the body and its development. And even to study the diseases, these transgenic animals are used, how genes contribute to the development of disease. So especially they act as models for the studying human diseases, and they help us to treat diseases by using this one. Today transgenic models exist for many human diseases like cancer, cystic fibrosis, Alzheimer's disease, rheumatoid arthritis, etc. So even these transgenic animals are very useful for production of biological products such as, which are very useful for us, like alpha-1 antitrypsin, human protein which is used to treat emphysema, as well as increase protein production in the transgenic animals like transgenic cow, as well as vaccine safety. So we need to test the vaccines before on these transgenic animals, and then if it is safety, then we can use it for human beings. Like that, even the chemical safety testing also, these transgenic animals are used, like toxicity, safety testing, and these transgenic animals are made to carry genes which make them more sensitive to toxic substances. And so the toxicity can be studied in these animals by giving them within a short period of time we get results. What is mutualism? Explain any 4 examples of mutualism. So mutualism is the interaction between 2 organisms of different species. Both partners will get benefited here. Example, lichens. It is a symbiotic association of algae and fungi, wherein algae provides the food for the fungi and the fungi in turn gets benefit by supplying the nutrition to the algae. Rhizobium and roots of legumes. So rhizobium is a bacteria which lives in the roots of leguminous plants forming root nodules. So the rhizobium bacteria supplies nitrogen to the plants and the plants in turn gives the food for this bacteria. Mycorrhizal association, as we discussed earlier, the fungi supplies phosphorus to the plants and the plants in turn provide nutrition for fungi. methanogens. So they are present in the gut of the ruminants, where methanogens get the place to live as well as shelter, food in the ruminants, but ruminants also get benefited by digesting cellulose from their food. So last bit of the question, where you have to answer only one from the given three. So identify the genetic disorder. Sex-linked recessive disorder occurring in about 8% of the male, only about 4.4% of the females, leading to defect in the cone of eye. So here this is color blindness. Color blindness occurs more in the males than in the females, as it is X-linked recessive disease. As males have only one X, there is a chance of having the disease more. So another one, sex-linked recessive disorder, single protein involved in clotting of blood. It is none other than hemophilia. Inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine, which converts later into other derivatives. So the disease is phenylketonuria. Autosome linked recessive blood disease, substitution of amino acid, that is at the 6th position, glutamic acid is replaced by valine. It is sickle cell anemia. Autosome linked recessive blood disease, rate of synthesis of globulin chains of hemoglobin is affected or reduced. It is thalassemia. They have given a flow chart here about the replication of retrovirus. You have to identify these 1 and 2. See, a retrovirus infects normal cell. It introduces its RNA into the cell. By the reverse transcription enzyme, it synthesizes its new viral DNA, because reverse central dogma. So RNA will be produced into DNA, DNA into RNA, and then protein. So the first one becomes synthesis of viral DNA. So viral genetic material in nucleus of host cell incorporate into host genome, and then second one, new viral RNA is produced by the infected cell, and then virus comes out of the host cell and new viruses can infect other cells. The other questions which are asked on this is, give an example for retrovirus. So the virus which is having RNA as genetic material, that is HIV. Name the disease caused by this retrovirus. HIV causes AIDS. Name the diagnostic test for this disease. It is enzyme linked immunosorbent assay. Give reasons for the following. Restriction enzymes are called as molecular seizures because they cut the DNA at specific sites by recognizing sites called palindromic sites. DNA ligase is used in genetic engineering. So ligase is the one which join the cut ends of the DNA, providing 3'5' phosphodiester bonds. Chilled ethanol is used in DNA isolation because for purification, precipitation, we require chilled ethanol. So lastly, after adding different enzymes, we digest cellulose, other type of macromolecules. So the DNA is precipitated and purified by adding chilled ethanol. Few DNA fragments move farther in a gel slab. Gel electrophoretic slab is used for separation and purification of DNA fragments, isolation of DNA fragments. So here, smaller the DNA fragment, farther it moves. So because of the smaller fragment size, they move farther. Thermostable DNA polymerase is used in polymerase chain reaction. We know that during PCR polymerase chain reaction, high temperature is induced wherein the two DNA strands get separated, and then it requires a special type of DNA polymerase which works at high temperature. We take it from Thermus aquaticus, that is Tuck polymerase, which remain active during high temperature induced denaturation of double stranded DNA. So this completes the model paper 2, which is given by the board, Karnataka board, and hopefully for next video we will be having paper one model paper solved. Thank you.